Hi, I'm Miss Nickerson.
No, Mr. Radford.
Welcome to the Viking Science Academy. Today we're going to talk about genetics and heredity. This is one of my favorite things to talk about.
It is.
Because it's applicable to you guys today. So when you have kids, it'll be your genetics. So let's talk a little bit about the history of it. What is genetics?
It's a study of our genes and how those genes are passed down to our offspring.
Okay, and we call that inheritance, how we get one thing from your parents and they got it from their parents and so forth, generation to generation. That comes from our chromosomes and our DNA, which we've been talking about, how they hold that code. And that's found in the nucleus, so don't forget that. Who's Gregor Mendel?
Man, he's the guy that kind of kicked off the father of genetics. He was very meticulous in what he did, and what did he study?
He studied peas, which sounds kind of silly, but peas work really good because you can find peas lots of places. They reproduce really fast. You can get lots of different plants to grow in a year, and they're really obvious on their traits, so like their color and their length and that kind of stuff. And he was a monk, and he just kind of did it in his spare time in a greenhouse on the side of the church, so it's kind of cool.
He used his data very, very well to figure out some of these laws that are associated with genetics that we know now. So
he was meticulous, is the way I would say. He really took really good, accurate data for his experiments. About 200 years ago, and he ran a couple experiments, and he found that he came up with two different hypotheses based upon his experiments. He found two things that he thought were going to kind of happen no matter what he did. And the first was inheritance. And that means that he thought that parents passed on something to their children. This was before we were looking at cells or knew anything about DNA or anything else. He figured out that something was getting passed on. We call those genes today. And so he came up with some kind of terms to describe that. So what's dominant and recessive?
Dominant is just a trait that would express itself. Think of someone that is like a dominant basketball team. They just control it, the game. Recessive, they kind of more, it's there, but it's not shown.
It's hidden. Okay, all right, gotcha. So these come from alleles, right? The alleles carry these traits on them, and they carry two. So I carry two different traits for eye colors. I carry two different traits for hair colors, but only one of them is expressed, and that's what we see, my brown hair and brown eyes. Okay.
One other thing with his background, he was a mathematician as well.
Yeah.
And so he used math to be able to kind of prove some of these things to be true. And so one way is using Punnett squares.
Yeah. And so Punnett square is just checking out probability, which is the chance that something will occur. It doesn't mean it will occur. It's the probability. So if you go to Vegas and play slots or 21 or roulette, you're playing the probability that it's going to land on black, or the probability it's going to land on red. It can land on black 20 times in a row, but theoretically it should land on each about 50%. Yep. Okay, so all of these types of things we're talking about on genes are called genotypes. So we're going to also use another word later called phenotype, but when we're talking about the what's on the genes, on the DNA, it's a genotype.
Yep.
Okay.
And then a phenotype is how that is expressed. It's physical appearance. So that pH, physical, that's how I remember that.
Oh, that's a good one. I hadn't thought of that. That's a good way. So I carry the genotype for brown eyes and blue eyes, but my phenotype is brown eyes because that's what I look. Okay, cool. So we chose the garden pea because they come in a lot of varieties. If you ever have seen peas before, their flowers come in lots of different colors. There's different shapes of peas, different heights, offspring, that kind of stuff. And he made them mate. So he cross-pollinated. He'd bring pollen from one parent and pollinate another one, and then wait and see what happened.
Yeah. Some very controlled experimentation.
Yeah, that'd be hard to keep all those, make sure that no bees were in there to pollinate his plants without him knowing about it. So what he found is when he took the parents, or the pea generation, and crossed two completely different genotypes and phenotypes, that they would have offspring. But then when he went into the second generation of offspring is where he started to see some of those characteristics come back out. And again, you said he's a mathematician. He came up with a formula, and then we represent that formula in the Punnett square. And when you cross one gene, that's called a monohybrid cross, mono 1. We're going to do a mono and a dihybrid cross, and we'll cross two genes. So here's a typical experiment.
Yeah. So P generation are the parents, the filial, the F generation are the first offspring, and then the F2 is the second.
I think he did like 20 generations.
Oh, it's crazy. I think you have a slide that shows how many he did.
So what he was recording and keeping track of were these characteristics in the pea plant. So their pods, their shape, their flowers, how tall they were, and then he would just marking them all out.
Yeah. And one thing, real quick, if you look at like the height of the plant, he crossed a tall and a dwarf plant. Some of us might think, well, their offspring might be midway.
Yeah, halfway, average.
And he wasn't seeing that, and so that's why he started to think, oh, something is dominating over the other one that's expressing that.
Exactly.
Yeah.
So he came up with some ratios that were, there we go, selected, and he was able to take those ratios, use his math mind, and come up with a statistical pattern. And that's what geneticists look for, statistical patterns, so they can figure out what are dominant and recessive traits, and then they can figure out how to manipulate those. Yeah. We've been breeding stuff for a long time.
Yes, we have, yeah.
So we breed cats and horses and dogs and everything else, and sometimes we kind of breed people too. So how can you keep one trait so strong through multiple generations? You guys may know some of these actors. These guys are all fathers and sons related here, and they have a really similar facial pattern and everything else. Some traits are dominant, and they carry through. Yeah. So tell me about these chromosomes again.
So homologous chromosomes, there's one of a matching pair of chromosomes, one inherited from mom, one inherited from dad. So
that's why we only have 50% in our sperm and eggs, right? So when they come together, we get the 100% that makes us up. All right. And then those chromosomes make a copy of themselves once they become part of you. And now I'm carrying 2 complete sets of chromosomes. One of them is going to be what's reflected, my phenotype, but my genotype is both of those sets of chromosomes that I'm carrying. They're called sister chromatids. All right. Sometimes we do get a blending. There was a hypothesis of blending, which would mean like, well, let's take this person's brown eyes and they mate with someone that has blue eyes, and their children will kind of have brown blue eyes. But that doesn't really happen so much. And so this was around for a little while, but like you said with the pea plants, when we bred a tall with a short, we didn't get a medium. We kept getting a tall, so there was no blending. So this was not a hypothesis was able to be proven.
Yeah, and these are actually some of these genes that we get, they're found on multiple places, so
affected by multiple breeding.
And we'll talk about that later.
So that led to the law of dominance. This is kind of one of those biological laws that we have a lot of evidence to back this up, which is when you put 2 organisms together, only one version is going to appear. It does not blend. We're going to have a dominant trait. We're going to have one set of traits that is expressed.
So in our example here, purple is most likely the dominant trait.
Yeah,
it is.
Exactly.
So, yeah.
All right, and then when we did this, we could go through a second generation, and we would see some of those recessive traits appear again. So we knew that they were still carrying recessive traits, they just weren't expressed. And that's what makes up our whole model of inheritance, that you still carry traits, but only your dominant traits are expressed, or the traits that you have. So alleles are for a very specific trait on a chromosome. So for example, this could be the allele for hair color, or the allele for if you have a monobrow, or freckles, or whatever. And sometimes they're in several spots, but most of the time they're just in one spot. Genes are for located all over on the chromosome.
The whole thing,
And we've got dominant and recessive, and we talked about both of those. If you have one dominant, they're a loudmouth. They're going to be what's expressed. And the only way you can see a recessive allele trait is you have 2 of them together, then they're powerful enough to kind of express themselves. So you need to make sure you guys know the difference between dominant, recessive, homozygous, heterozygous, and we'll get to those in a second here. So Punnett squares, little tic-tac-toe kind of.
Pretty much, yeah.
Or battleship, as I call it, the genetic battleship. This is a monohybrid cross. How do we set it up?
So we take we split up the genotype, from mom on top and from dad on the side, and then where we take, it is battleship. You take one and you match them up in each of those boxes. We'll be practicing these quite a bit, so it'll make more sense.
And you split them up because they can only pass on one, right?
Through
their sperm or their egg. They don't get to pass on both of them. And so then we see the genotype expressed with letters, and we use capital letters to represent dominant, and lowercase letters to represent recessive, and then phenotype is what it actually results in. Now these guys use P for this. I think that's a really bad letter to use. If I was going to use an uppercase and lowercase, I would use letters that look totally different. So maybe like Q would be a great letter. My handwriting, P's wouldn't cut it. And so we can do this with offspring for sex and sperm also, and we'll be doing this in our second lecture on sex-linked genetic traits, and this is a little bit more difficult, but for now we'll just do it with a plain old Punnett square and traits. And we can see there, if they have two of the exact same alleles, so dominant, we call those homozygous. And if they have one of each, we call those heterozygous, like heterosexual or...
So, yeah, you could have homozygous dominant, homozygous recessive, and then there's heterozygous.
And then heterozygous, because that means one of each.
So in our example, the homozygous dominant would be two big P's on here. Homozygous recessive would be two little p's, and then heterozygous would be a big P, little p, one of each.
Okay, so you should be able to in class to take two sets of parents, cross them, and then get offspring, and then take that offspring, cross it again, and get a second generation of offspring, and be able to tell us what they look like and what their genotype and phenotype are. So here's an example using the letter A. You can see how we're moving down and across. Why don't you take a second and pause the video and see if you can complete this test Punnett square, and then afterwards tell us how many will be dominant and how many will express recessive. And we're crossing a homozygous dominant with a homozygous recessive. Okay. Hopefully you got that it's going to be about 50% of each, and they're all going to be dominant because we have dominant, but one of them is going to, they're all going to, 100%.
Yeah, on that one they're going to be 100%.
And all of the offspring is going to be heterozygous. They all had one capital T and one lowercase t. Now, just as we learned in DNA, there's other factors too. What are some of those other factors?
Well, I think some of this goes into the epigenetics stuff. So our environment can change our what happens to us. So.
And hydrangeas, which we don't grow too many of them in here, but their color depends upon the acidity of the soil. So even though their DNA is the same, that environmental factor is having an effect on how it's expressed. So, all right, so we'll see you guys in class to do Punnett squares and practice our vocabulary. Thanks for joining us for the Viking Science Academy.